So I kind of have a modular talk, so I'm going to take my time, and if people have questions, I'm happy to answer questions as we go. And then if we're running out of time, I'll just drop the last section of the talk. So it will be totally fine, don't worry. So, and I have 3 hours, right? All right. I first really wanted to focus on this question of, especially since we're here at the hospital at the program that has a very strong clinical component, we're all sort of thinking forward, or I guess maybe if we're very optimistic currently, about precision medicine. And I wanted to ask this question of, so why do we actually need precision medicine, and do we need to start thinking about whole genome sequencing, right? Because all of these billboards for all sorts of hospitals, including, I'm sure, Presbyterian, are telling us that they're going to sequence your entire genome, and then they will do precision medical treatment of disease X, whichever one they're advertising at that particular billboard. But how much of the whole genome are we actually using and thinking about, and to which extent can we actually look at it, and is it actually something that is really going to be helpful? So how do we interpret the impact of the regulatory mutations? That's really probably the key, well, I think the key question when you start thinking about whole genomes going beyond exons, right? Because when we're talking of the 98% of the genome that's outside of the genic regions, for the vast majority of these mutations, we actually know very little about them, right? At best, we might be hitting a known transcription factor binding site, or we might be looking at some sort of cheap-seq or other data that functionally tells us that maybe something is going on in that region of the DNA. But how can we actually interpret it with precision, where we can say that this particular A to T change in the DNA is either not functional, or biochemically functional but benign, or biochemically functional and increasing your risk to predisposition, let's say, to Alzheimer's by a certain fraction. So that means that we need to be able to, at the biochemical level, predict a precise impact of any mutation anywhere in the genome, right? And when we're talking about these regulatory mutations, then for the most part, if we're talking about either effect on transcription, so that means that they're affecting either, let's say, transcription factor binding, histone marks, chromatin accessibility of the DNA, or we're talking about post-transcriptional effects, right? So this is, while I'm talking about regulatory mutations, these might be mutations actually in the genic region of the genome, right? But the effects that they're having are not so much affecting the coding sequence or the structure per se. What they're affecting is, for example, splicing or stability of this RNA, right? So they're really going to affect, in a large extent, having their effect through the RNA binding proteins, right? So that's the post-transcriptional effects of these regulatory mutations, right? And that we need to be able to have a precise effect predicted for any variant, and that includes variants that are not at all common, right? Very rare variants. And by that, I also include variants that we may not have ever seen in the wild, right? We may never have seen this previously in the genome, but we need to be able to know what the effect will be. So how does a single letter change in the genome impact gene regulation? Then we need to be able to say, past this biochemical effect, what does it actually mean in terms of if it's a transcriptional effect, gene expression, right? So we don't want to just be able to say, oh, it affects GATA1 transcription factor binding, and it decreases it, the end. We need to be able to say, and that means that this particular gene will be expressed this much less in this particular tissue, and eventually, of course, cell type, right? And then we want to be able to know how do these gene products actually interact, right? And finally, how they're linked to actual human disease, right? So which ones of those mutations are going to be disease causal? Because just because they're functional biochemically doesn't necessarily mean they're deleterious, right? And so how are they disease causing? How do they affect protein interaction networks? And by that, I don't necessarily mean the physical, if you're from the bioinformatics end of things, I don't necessarily mean technically I really just mean pathways, right? How do they affect these complex cellular networks? How does having less of that particular gene product affect that pathway? And how does that pathway link us to a particular human disease? So I'll tell you a little bit about, before I fall over and kill myself here, I'll tell you about some of the specific deep learning models that we've developed to answer these questions. So DeepC, Seqweaver, and Expecto are deep learning models that predict transcriptional for DeepC, post-transcriptional for Seqweaver, and actually expression effects of mutations based purely on sequence, so not learning from mutational data, and therefore they can predict it for rare variants and those that you've never seen before. I will tell you about functional networks. So this is methods that we've developed for integrating thousands of largely expression data sets, but really I call them officially the kitchen sink methods. So this is, we integrate any data sets that might tell us about how proteins are expressed, regulated, and interacting. So it's physical interaction data, expression data, information about transcription factor binding sites, potentially protein domains, although right now we actually exclude that because of circularity issues, right? So anything that could tell you something about what proteins are doing in cellular networks, but it is vastly still going to be expression transcriptional data for now. And then finally, if I have time, I would love to tell you a little bit about, so Humanbase is actually our large site that has all of these networks, all of our deep learning predictions, all accessible in an interactive interface. I'll just show you a few screenshots throughout, and also you can, for this group actually perhaps more interestingly, you can download all of the predictions as well. Like for example, these full networks if you want to use them as input into other methods. But I might be able to tell you a little bit about DiseaseQuest, which is our system for really integrating model organism data with human clinical disease information to try to make better predictions and really experimentally tested predictions of human disease-associated genes by really identifying which particular aspects of model organism biology are relevant to a particular human disease, so that you're not just sort of making predictions, for example, for, as we actually done there, for Parkinson's disease genes, then testing it in worm, and then hoping that this works out, and maybe these are conserved areas of worm biology, but you actually are making predictions specifically zeroing in to the part of biology that is conserved in worm, but associated with Parkinson's disease, right? And so sometimes you might get no predictions, and that means that maybe worm isn't the best model system for that disease. But if you are getting predictions, they're really zeroing in to the aspect that's relevant, and then you can test them and then feed it back to human biology, and now you actually have experimentally verified potential candidates for human causal disease genes. All right, so without further ado, let's think about how do we actually use these whole genome sequences, and why is it hard? I think for this audience it's probably obvious, but I really want to emphasize this. It's so hard because, of course, all of these folks, the so well-drawn, multicolored people on there, might actually have the same exact disease, but their underlying genetics might be completely different, right? And a lot of it includes rare variants that even if we sequence huge cohorts, we're just not going to have realistically enough power to be able to have enough statistical power for every rare variant, as well as variants that might actually we never even see or see in very specific context survive past basically fetal development, right? But we're not going to be able to analyze that and have the power to be able to identify them. So that means that we need to be able to associate mutations with outcomes without ever relying on mutational data. We really need to essentially go from purely looking at quantitative genetics, which is, of course, a very important way to go, but have a complementary way of looking at actually essentially developing a regulatory code in the same way that we had for the coding variants, right? So all of you have taken basic biology at some point. So this, there it is. So this, some form of this code table, right? The coding variant, our regular code in every molecular biology textbook, genetics textbooks. I don't know where they actually study this at this point, right? So you can look up if you change a T to a C in this codon, it does not affect the amino acid, right? And you can literally go through and figure this out. I'm oversimplifying this a little bit. Those of you who work in this area, of course, there's missense variants and a few other things that are still left to be discovered. But for the most part, you can actually go and look in this biological code and figure out what effect each mutation has. What we need is an equivalent of that for non-coding variants. And by that, I don't necessarily just mean variants that are outside of the genes, but basically, I mean non-coding effects, right? So either they're effects on splicing, effects on RNA stability, effects on regulation and expression, right? And so we need to be able to, for every variant, be able to say what exact effect it will have biochemically. For example, this particular variant will reduce GATA1 binding by this much, or it will affect this particular histone mark. So that's actually what we set out to do with DeepC and seqweaver. So DeepC, can I use? Nope, this is not working. OK. So if we have, I'm not telling you how the system works. I'm going to tell you that in a second. But I'm going to show you how you can apply it once you have the trained model, right? So the idea is that once you train the model without ever having seen any mutational information, now you can give it a mutation. So for example, here's a variant from A to C change. You throw it into these deep learning models, DeepC for transcriptional variation prediction, seqweaver for post-transcriptional. And you're going to get specific for that one specific change, specific effects for thousands of histone marks, DNase accessibility predictions, effects on various transcription factor binding, as well as effects on RNA binding, protein binding, right? So that means that, for example, in this mythical A to C change, perhaps in the A variant, GATA1 transcription factor bound really well, whereas in the C variant that we see, let's say, in an autism patient, now the binding is very strongly affected, and we have a much reduced GATA1 binding. So perhaps that would be a prediction for us that may be one of the variants that may explain the autism predisposition in this patient, right? So this is what those systems do once they're trained. But how do they do it? So the way that you train these models is you actually never give them mutational information. You just give them one reference genome. Well, I mean, any human genome. In our case, we actually do literally use the reference genome. And then we give it lots and lots of vectors of feature information. So that, in our case, is ENCODE, CheapSeq information, and roadmap epigenomics CheapSeq information. So that means you basically just have long runs of, for example, I'll just stick with GATA1 as my example, of peaks of where GATA1 was found to be bound in certain cell types that ENCODE or roadmap epigenomics has looked at. Same sort of peaks for various histone marks, DNA accessibility experiments, et cetera, right? So we have the original system that was published, DeepC, in 2015, had 900 different features, so 900 various transcription factors, histone marks, et cetera, effects. Then the systems that I'll talk about in a second that we applied to autism data have over 2,000 various features. And what you do is, so it's kind of, how does it learn, right? Because it never sees the mutational information. So the way it learns, just the intuition of this, is that it's actually learning along the genome, right? The reason that it can actually learn what it means for, what is the sequence signal for, let's say, GATA1 binding without knowing anything about GATA1, its transcription factor, binding sites, et cetera, is because there's multiple sites where GATA1 is going to bind within the 3 billion long letter sequence of the human genome, right? So it's actually learning as it goes along the genome, as opposed to as it looks at many genomes, right? Because it only has one genome that it's looking at. Does that make sense? And it actually gains a lot out of the fact that at the bottom, right, the convolutional layer, that's what it's doing. It's really scanning for these motifs. And then the pooling layer, can you guys actually see any of this? Unfortunately, when I look at it from the side, I can't see anything. You're seeing it, right? All right, so I won't point because I actually can't tell where I need to point without standing right in front of it. So the pooling layer is actually going to spatially scale it, right? So it's actually going to be able to take into account higher order sequence features in the next convolutional layer, right? So that's really taking into account these features that we know should be important just from our knowledge of biology, right? We know that various parts of DNA should be actually informative for each other. And then an important point here is that in addition to capturing this context information in the hierarchical structure of the convolutional neural network, it's actually able to capture interaction of different sequence features. And by that I mean the fact that perhaps certain histone marks are actually predictive of, let's say, GATA1 binding, right? And that you will have a certain chromatin accessibility conformation if the transcription factor is actually able to bind, right? So this multi-layer nonlinear transformation on the top is what is actually capturing interaction of these sequence features. And finally, at the end, we actually do multitask prediction of these chromatin features so that we are actually integrating all of these different sequence features as well as across all of these different cell types to be able to achieve better prediction. So it actually works pretty well. If you're interested, I will pull up the slides with the AUC curves and all of those evaluations. But I want to actually get to the applications of this model. And it's been published for a while, so I just stuck some articles. People have been using it quite widely. My favorite one is, they've been used in polygenic gene list, cancer, et cetera. But my favorite one is the eyebrow thickness. It's the one on the right in PLOS Genetics. So I always highlight that one. All right. So does this make at least some sense? I don't want to lose anyone at this point. If this is good, then I want to show you how we can actually, so if we have this model, right, that can tell us whether a particular regulatory variant is disease-associated or not, and what are particular biochemical effects of it, then we should be able to actually use it to analyze real clinical data, right? So, and this is data from a large study of autism. So autism, I think I don't need to introduce and probably don't need to really tell you how important it is. It's, there's all sorts of debates on to which extent it's rising, but it's highly prevalent. It's 1 in about 70 kids is diagnosed. It's highly heterogeneous. It's very challenging genetics. Even the mutations that we see most often in the coding regions, this is still very small fractions of cases that we see these mutations in. And this is a collaborative project together with, so my former student and then postdoc Jian Zhou, who is now a faculty member at UT Southwestern, who developed DeepC, and Chandra Tesfield, who is an experimentalist, a wonderful experimentalist in my group, with Chris Park, who is now a research scientist at my group at the Flatiron, before was my grad student, but did this work actually while a postdoc in Bob Darnell's lab. Bob is at Rockefeller, and then also Alan Parker at the Simons Foundation, who really was our autism expert. And we were motivated by this fact that there's been a very large open question looming in not just autism, but in general in pretty much all of complex human disease, where there's a lot of evidence that seem to be pointing to importance of regulatory de novo mutations, right? So by de novo, I mean mutations that your parents don't have, but you get. So, things that it's genetic causes, but it's something that you don't inherit from your parents. And there's been a lot of evidence that we're sort of partially pointing to that these regulatory de novo mutations seem like they should be important in various areas of human disease, and not a single one have there been able to be a proof of this. So in fact, about 6 months or so before we came out with our paper, there was a paper, so this one is actually from a month before. There was previously, about 6 months prior to this, a paper in Nature Genetics that said in the same exact data that we use, this large study of autism cohorts where there's about 2,500 family, it's a quad study, so mother, father, and child that's affected, so proband, child with autism, and an unaffected sibling, all whole genomes. So they looked at that same data, and they said there's no way to show noncoding effect. You need to have at least 8,000 families to be able to have any hope. And then about a month before we published, this science paper came out that also showed that this is basically them, the obvious thing they can say. So the way you do this in quantitative genetics, by the way, the way you show the effect is you literally count how many mutations, non-coding mutations you have for the probands, for the kids with autism, versus how many mutations do you have for unaffected siblings. And if there's a statistical enrichment in the probands, then you have your proof that quantitative genetics is showing us that there's significant effect of non-coding mutations in the kids with autism. Because the siblings are the strongest, really, well, short of identical twins, right? Like this is the strongest or control you can actually have. So that didn't work. So maybe the next smartest thing to do would be you can overlay over these and say, well, I don't want to just count the mutations. I want to say, well, I'm only going to count the mutations that are next to autism disease genes. Or I will only count mutations that are, in the peaks, cheap seq peaks from ENCODE data, for example, right? Or I will only count mutations that are in known transcription factor binding sites. So this is what that figure is. So this is that science paper where they tried every one of those options, literally, anything. That's why there are so many dots there. And you can see that none of those dots are even close to the significance cutoff, right? Because basically, once you do so many tests, you have to control for multiple hypothesis testing, and you're not even close. So what we said was, well, but we have this model, which yep.
Was it Bonferroni-based?
Yeah, it's Bonferroni-based, but honestly, actually, even if you do correlation, any of the others that take into account, you're not even close. Basically, they can't, right? Like, look at how far.
You use FDRs.
FDR doesn't work either in these with the overlap. It's just it's just so many overlaps. Yep. I think actually that might be. No, this is a p-value. We have tried the FDR as well, and it doesn't help in that one. So basically, we're going to just do one simple thing. We're going to, instead of doing all of these overlays, we're going to use the DeepC and seq-weaver models to predict the effect of these variants. And so then you get a single score, disease impact score, that tells you how likely to be disease-causing are these, right? And of course, this knows nothing about, it didn't see any of the autism data, well, other than the predicting variants. So what we're looking at is, to prove this, we want to be able to say, well, the siblings have a lot lower, significantly lower disease impact score than the probands, right? And there's no multiple hypothesis test because you just do this one test. And indeed, we see that both at the transcriptional, I do really want to show here, sorry, both at the transcriptional level and non-transcriptional, post-transcriptional level. So these models are separate. They're both deep learning models. You actually see significant difference between siblings and probands. And this difference becomes actually larger when you look at variants nearby genes, or if you look at variants nearby genes that are loss of function intolerant, right? So this is not the LOF intolerant is, of course, the definition that was, that's the exact consortium where they look at conservation, right? So these genes are much more likely to be disease-causing genes, right? Because they're conserved and not tolerant to loss of function. So this actually was the first proof, not only that non-coding effects in de novos are really real in autism, but actually for any human disease as well. And this is not just sort of this generic signal. You can actually look under the hood of the models and see where you see the biggest difference. So a lot of these models are just from tissue culture experiments, right? This is where ENCODE data comes from. But we do have the GTEx consortium data as well, which I know many of you are familiar with, since some of the members of this program are members of the consortium. So if we focus on the GTEx tissues, we can actually see for which of the models for tissues is there the biggest difference in signal between proband and sibling. And so here we're just mapping these models purely by that difference. And you can see that basically the top, I can't remember, 12 or so are all brain models. And everything that's significant is brain. And then there's like a couple of brains that are right below the significance cutoff. So it's not just that this disease impact is really giving you a signal, it's actually quite tissue specific. You can actually look at all of these predictions if you'd like at Humanbase. So you can, if you're interested in autism, look up at any of these predictions, look specifically what the features are and how they're predicted. So you can see there particularly it's highlighting the fetal brain tissue and what the specific feature, the histone feature that's being affected, and it's predicted to decrease, for example, in that case. But we actually wanted to test some of them experimentally. Now, of course, this does not prove that they're truly driving autism, right? The autism connection is really coming from this quantitative genetics argument. But can we actually prove that this is actually driving transcription, right? So this is both verifying that the deep learning models work and hopefully convincing you that at least it's really finding real biology. So we took, I can't remember, I think this is around 40 various transcriptional effect predictions that were predicted. These are not the top 40 because we basically wanted to pick some that were around. We really wanted to test, various things that some of them are really close to the promoter, some of them are distal enhancers. We wanted to test some genes that are very well known to be associated with autism, whereas some of the others that weren't. But what I can tell you is that we confirmed that in tissue culture, if you actually look at these fluorescence assays, and you literally make a version of the driver that has the proband allele, versus the single variant change that the sibling allele has. So these two, the two bars here have only one nucleotide difference between the proband and sibling. In this luciferase reporter assay, 94% of the time it actually does drive significant differential expression. And then what was interesting, actually, this is just an aside, but to me what was surprising, so I was very excited that it was so well verified, we were all excited. I was actually most surprised by the fact that we got about half of the ones that actually significantly decreased expression and half that significantly increased expression. Some of my transcriptional, colleagues from the transcription field said they were completely not surprised because they've been looking at enhancers and how there's a lot of evolutionary push to try to not have enhancers be completely optimized. I have to say I was kind of floored by this. I was very interested. And of course, this is a small set, so I don't know if this is a proof of the fact that, but we weren't picking them for half and half. It just so happened, and so I actually found this quite interesting. Now, I told you that we did, so this was transcriptional regulation. I told you that we also have this proof on the post-transcriptional regulation level. So here's an example of RNA binding protein disruptive regulatory mutation in a variant of SMEC1. And SMEC1 is actually not one of the proteins that's known to be associated with autism, but it is one of the very highly conserved proteins that's quite important, is neuronally expressed. And this particular mutation does, in fact, disrupt splicing of SMEC1. So here you can see that, if you're not familiar with the bands, it's probably easiest to just look at the bar graph. And you can see that there's a significant difference in the reduction. Basically, this particular ASD proband-specific mutation causes a significant reduction in the long isoform of SMEC1. So it actually affects not the level of SMEC1, but actually splicing of SMEC1, specifically in this kid with autism. And interestingly, for these mutations, so this is not for SMEC1 in general, but in general for these RNA binding protein disrupting mutations, we actually found that higher dysregulation at this post-transcriptional level was associated with significantly lower proband IQ. So among kids with autism, so this is not in general population, among kids with autism, if you have more, for kids with autism that have more mutations that affect basically splicing, processing of RNA, stability of RNA, essentially how RNA binding proteins handle RNA, those mutations specifically cause lower IQ because, of course, autism kids have very wide range of IQs, and some of the kids have very strong social impact, but actually close to normal, sometimes higher than normal IQ. And this was a very interesting observation. And you can see that it's actually highly statistically significant. So that was the story of applying actually these deep learning models to a real quantitative genetics clinical cohort of autism kids. We have since, in collaboration with Bruce Gelb at Mount Sinai, also applied it to congenital heart disease patients as well. And so there, it's not a quad study, it's sibling. We actually use the ASD siblings as the control, so it's a little bit more complicated, but we see very similar. So basically, we see a proof of de novo non-coding mutations having an impact there for the first time as well. So that's in revision right now. So that it seems to be that these models really are quite useful in being able to analyze whole genome data in at least this, I guess, clinical research, quantitative research setting. And hopefully in the future, I don't think, by the way, just for full disclosure, I don't think these models are where we should all be running and clinically using them the second, but I do think we're starting to get to the point where we can start at least thinking of how would we integrate various non-coding, coding, as well as clinical sources of information in the future in these actually making clinical precision medicine decisions. We're not there yet, aside for certain research scenarios or scenarios where there's just no other choice. But if we don't start thinking, I think this is the time that we need to start thinking about it, or we're going to sort of be catching up to the bus, or whatever is the best analogy. All right. So I want to then sort of continue on this and go beyond being able to predict the disease impact or the biochemical impact of these mutations to being able to predict tissue-specific gene expression purely based on sequence. So can you, my extreme example is if you give me Neanderthal genome can I predict to you how exactly genes would be expressed in Neanderthal purely based on the sequence, upstream sequence in the regulatory regions of these genes? And Jen actually has done that, although we don't know exactly how to test it. We don't have any Neanderthals around, so we haven't yet published it. But I will tell you about how the system works. So this is just showing you the example of what we really want to be able to predict. So here we're putting in CACNA2D3. It's actually one of the genes that does seem to be quite important in autism. And you can see that what we're getting is predictions. So on the top there, on the, the blue numbers are all the different SNPs variants that we see, and we can actually predict the effect of these variants across different tissues on the expression of this particular gene. And just to be clear, it does not mean that every single variant in these regulatory regions is affecting expression CACNA2D3. Just for visualization purposes, we don't show any variants that don't predict significant change, right? So these are only the ones that do predict something, or it would be just a very long scroll bar. And in this particular case, so it turns out that we actually did verify one of those, and it does cause a significant expression change, again, in luciferase reporter assays. So this is work, again, by Jan Zhou and Chandra Tesfield, and the Humanbase, the interface and all of that was actually great work by Aaron Wang, who is the head of the Humanbase project at my Flatiron group, and Kathy Chen and Kevin Yao, who worked with him on this visualization. So how does Expecto work? The goal here is really ab initio prediction of tissue-specific gene expression for sequence. And I know I'm sounding like a broken record about this, not using mutational information for this, but this is really important because if we use mutational information, right, so the obvious way to predict effect of variants on expression is you take, let's say, GTEx data where you have actual sequence, right, genetic sequence and matched expression levels, and then you build your favorite classifier to learn which variants cause which changes in expression. And the problem with this is, it's great. It's not a problematic approach. The key challenge with this is that then you're not going to learn anything about rare variants. You're only going to be able to predict the variants that you already saw. And I would argue that a lot of the variants that are associated with disease, if not the majority of variants that we really need to know about, might be rare, or at least rare enough that we're not going to be able to see it regularly enough to learn about their effects, right? But, and also, the other question with expression is you really want the causality to be clear, right? You want to be able, so if you just use cheap-seq data to predict expression, that's actually another very helpful way to go, and it's a very useful approach. But there is a challenge there of which way the causality is going, right? Because of course, changes in transcription factor levels can cause expression changes, but expression changes are things that change transcription factor levels, right? So here we actually now have a basically in silico cheap-seq, right? These deep C model is what can, given a sequence, actually just predict for you what are the levels of different transcription, right? What is the binding effect going to be on transcription factor binding? What are the different histone marks going to be like, right? So we can actually go purely based on the sequence. So we're going to take sequence around the TSS, so the transcription start site of every gene. We actually went 40 kilobases, so 20 upstream, 20 downstream. We did actually look at whether going further out or shorter in. So shorter in, you do lose some information. Further out, it doesn't help that much. I would not conclude that that means that there's some secret information source only there. I do think there's a lot of information, as we all know, about transcriptional regulation right close to the transcription start site. But I think the jury is still out if the fact that we didn't get better performance going further out is just the fact that we don't have enough data to learn much further out, or if it's truly some biological conclusion. But right now, yep.
So enhancers further out.
Exactly. So there should be enhancers further out, and they should have an effect. So my guess is that it's really more the fact that right now the vast majority of this tissue-specific data that we're evaluating on, we're getting basically enough of accuracy for 40K that going further out, we're losing so much of the signal because it's harder to predict it. Exactly. So that's why I always want to make that claim, because I don't want people to start concluding that this is where biology is. I think this is where, given our current data, the signal is. Let's put it this way. So what we're going to do is we're literally going to run deep C across that region, and that allows us to predict cheap-seq peaks, right? So transcription factor binding sites, transcription factor binding events, histone marks, Chromatin accessibility, right? So this is basically like running ENCODE in silico. And of course, again, my other big disclaimer is this does not replace biology. The only reason we can do this is because all of these experiments have been done, right? So just, I really want to make sure because, well, people make the wrong conclusions, but also I don't want this, I really want to make sure that these projects get funded because that's the only reason we can do these models, right? I think we all recognize this. So that means that then we actually are just going to take this quantitative in silico predicted cheap-seq peaks. And now we're just going to make a very simple supervised model of that cheap-seq peak trained with GTEx data, right? So another wonderful large public project, right, where we have expression for a number of tissues. So this is 230 or so tissues from GTEx. And we actually just use regularized linear models. We just wanted the simplest model so that the overfitting likelihood is the least. But you can throw in your favorite model here. And then, oops, sorry. And then what you're going to do is you're actually going to end up with the tissue-specific gene expression prediction for each gene in each tissue of those GTEx tissues, right? And then that means that also if you have a variant, you can actually predict the impact of that variant on tissue-specific gene expression, right? We've evaluated this actually quite systematically. The most obvious evaluation to quote is that you can actually take a chromosome, so this is how we evaluated DeepC as well. We keep a chromosome or two completely out of the whole process, so the system never sees that there exists. I think it's chromosome 18 and 20 something. That it never sees that there is this chromosome. It never sees any cheap-seq data, any genomic sequence, and then we actually predict for that chromosome. And you can be really careful and actually filter out any genes whose upstream regions have any similarity to any of the other chromosomes. That actually turns out to have no impact. But if you then take that chromosome, you can make predictions for tissue-specific gene expression of the genes on that chromosome, and that's your holdout set. And it turns out the correlation to real GTEx experimental data is about 0.84. So it's actually, I was quite amazed, quite impressive based on purely sequence. But before you get too excited, I do have to point out that if you take two different patients and look at their expression correlation, it would be about 0.96, right? So this is not yet. If I told you it's that good, you should not be believing me, right? Because there's no environment, right? We're just looking at 40K around TSS. It's purely what is in the sequence already, right? There's no time, there's no developmental information, none of that. So, 0.86, yeah, my guess is probably the majority of that gap is probably all of those things we're not modeling, but some of it is probably also just that the models aren't perfect. But I think a lot of that is probably just the stuff that's not even we're not even trying to include. All right. So then, and it is capturing tissue-specific information as well. You can actually look at the difference of each gene tissue-specific expression from its mean expression across tissues, and that correlates as well. It's not perfect, but it's definitely not just the easiest way to get good correlations, right? For the students among us, just be always careful where you're getting predictions, right? Because there's a real and good, from the computational perspective, very correct way where we would have this all held out and everything is done well. But if we just predict ubiquitously expressed genes correctly, we quite likely will get a really good correlation because those tend to be highly expressed genes. They tend to be ubiquitously expressed across, they're by definition ubiquitously expressed across tissues. So you can get a good answer, but it's not the answer you want, right? You want to see how you're actually getting tissue-specific expression right, and that is true there too. Now, what can you do with it? Well, one obvious thing you can do with it is you can look at GWAS variants and see if you can actually predict which of the variants that are in LD with each other, right? So if there's multiple variants in the region where you can't tell which one is causal, which is typically what you have with GWAS, can we actually, and what people usually do is they start looking at those variants and they say, well, this one makes sense because of this and this and this region, or I really think it's here, it overlaps cheap-seq peaks, et cetera. And so in here, we can just say, well, there's a variant that we actually predict will change expression of a gene. Here's one that's not. So we actually did this for 8 different studies in autoimmune disease. So in this case, it is immune-related diseases. So this is all our top 8 predictions. We just noticed in the for our immune-related predictions, there was a lot of disconnect between our very confident predictions from Expecto and the GWAS studies. What they said was their lead variants. So you can see that here's one example. For example, In the orange, our Princeton color, you can see that orange ball. That's the variant that Expecto says changes expression. In this case, it's this gene called IRGM locus. It's an important gene in immune system, and it turns out that it's been implicated in Crohn's, ulcerative colitis, and IBD. So there's actually five different studies, GWAS studies, across these 3 different diseases that all said that had their lead SNPs for these various diseases. And you can see those are the blue crosses. And it's a little hard to see, but you have to just trust me that the blue crosses, none of them overlap the orange ball. So essentially, Expecto is absolutely sure that the variant that's really the cause of this is none of the variants that was called across these 5 different studies. So we actually tested it experimentally. Now it's cell culture assays, but we can actually see which of the variants actually drives an expression change. And it turns out that indeed the predicted causal SNP by Expecto does drive differential expression, whereas none of the ones that were lead variants that were listed in these GWAS studies drive significant differential expression. And this was true also, so this is the IRGM locus. This was also true for this chronic Hep B associated HLA DOA locus, so same exact picture. And I have another story that I didn't put in the slide there, same picture as well. So in this case we did, so in other cases, right, we verified 94% were verified. In this case, for whatever luck of the draw, I don't think it's necessarily that we will always have 100% accuracy, but in this case we did, everything we tested did turn out this, with this. I mean, blood models are probably the strongest in Expecto, so this is not completely, I would not make conclusions that we can now outdo every single GWAS study perfectly, but this does give you a very powerful tool to be able to actually try to look for causal variants, right, by being able to figure out which variants actually affect tissue-specific gene expression. All right, I'm going to really briefly tell you about networks, and then I will skip the last section so that we would have time for a little bit of questions. So once you can predict tissue-specific expression, there's still a question of what these proteins are actually doing in the cellular pathways, right? And when we think about pathways, a lot of times we actually simplify them and think about functional networks. And by that I mean the big negative is we are losing the mechanistic biochemical information, right? So these are fully connected weighted graphs for those of you who really like specific definitions of it, of where we are looking for how likely are 2 proteins to work functionally in the same pathway with each other. So that means that a kinase and target, for example, or a transcription factor and its target, or 2 proteins that physically bind each other, right? So the power of these graphs is that they really give you a very nice summary, functional summary of huge collections of data that might tell you about how pathways work. And they allow you to compare across tissues and across organisms as well, because they're fully connected and weighted. And they have very high sensitivity and pretty high specificity. But the negative, of course, is you are not taking into account any of the biochemical, pathway-specific information, right? So a kinase and target versus 2 physical interacting proteins will both have just a connection that's probabilistic connection, right? We won't label them. And so the way we do it is we take thousands of genomic data sets, again, large extent expression data, and we want to make tissue and cell type specific networks out of them. And we want to be able to do it even for tissues and cell types that might not be well represented or represented at all in these connections, by essentially using hierarchically integrated tissue-specific knowledge. So by that I mean basically known pathways and information on which tissues and cell types as certain members of those pathways express. So the intuition is if you have a kinase and its target, and that kinase and target relationship is in your molecular biology textbook, that pathway probably doesn't exist as it's drawn in your molecular biology textbook in any tissue, right? It's an amalgam of many, many different tissues and cell types. But if the kinase and the target are both expressed in D2 neurons of the brain, it's quite likely that that relationship is happening there. So that's how we get the gold standard. And then we can basically look and weigh each data set based on their ability to recapitulate the gold standard. And then we just do standard naive Bayes framework that integrates across these data sets weighted by their ability to reflect the standard. And you can actually get fancier here by making it semi-supervised, because you can actually make predicted expression of kinase and its target. You can predict whether they're expressed in the D2 neurons of the brain. Or another thing you can actually do is you can use a different organism and see if you can use, for example, mouse data where you might have much better resolution, for example, for specific neuronal cell types versus the human, et cetera. So then you can repeat it for all pairs and predict these networks. And so you can download these actually tissue and cell type specific networks from our Humanbase website, or you can interface with them. And this is actually showing that just for one example where we look at IL-1 beta specifically, it's a very important cytokine that's important if you're studying cardiovascular disease and inflammation-related cardiovascular disease. So our collaborators were interested in what happens post IL-1 beta induction in blood vessel, and it turns out indeed our predictions are 18 out of 20 of those top neighbors actually do change at the first time point upon induction. Turns out the other two are correct, but they're just not transcriptionally regulated. But most importantly, that prediction, the most significant specific network in accuracy is the blood vessel network. And the cardiovascular networks as a whole are the next set of networks that are accurate. And if you look at the general network that's not tissue specific, it's not even close to the accuracy of the IL-1 beta network. And you can also look at higher level network architecture in human disease. I won't get into this, but this is looking at Alzheimer's, again, on Humanbase. So I am going to skip out of this. Whoa, didn't mean to skip out of this quite so much, sorry. And just go to the last slide. So I told you about a deep learning-based algorithmic framework, right, for predicting effect of non-coding variants in the genome, both at the transcriptional and the post-transcriptional level, and how we can actually use them to predict tissue-specific gene expression purely based on sequence, as well as variant effects on tissue-specific expression, how we can apply this to study complex human diseases, including autism, and finally, how we can actually use functional networks to understand that once these proteins are expressed, how they interact in different tissues and cell types. And I didn't have really time to tell you about disease quests. I apologize. I don't do anything anymore. I just go around and give talks and write grants and such, all the fun stuff. But I have an amazing group of students, postdocs, research scientists, and such who are actually doing the work. I think I've actually acknowledged everyone as we were going. But I just wanted to put up a slide with their pictures as well, and would love for you guys to check out our Humanbase. Also, if you're interested in any of the deep learning models, we actually have this Selene open source library specifically made for deep learning sequence-based models in biology. And the idea is really to make it very easy for you to train new models, for test new models, to develop new models, to share them. And it also interfaces with Kipoi and other, the Kipoi model zoo and other such products, but it really focuses on being able to actually train new models as well without having to, figure out how to load your sequences and do all the deep learning from scratch.